we couldn't have all of this without the support of GRDC, which currently sits to 1.5 million a year, plus all of these other projects like this one that is a 2-year type of project focusing on the resistant testing, because it's an issue that it's believed something that we want to ramp up, or anyway to take to a certain level that then is a sort of tool that you can use whenever you need it. And it's not just 1 year. This has been 2 decades, starting with Steve Powers and then continue with Hugh that we have here today. And all of the knowledge that has been produced now allows me to do the easy job, which is basically testing for resistance to a number of chemicals. But a lot of hard work is in the background, a lot of validation, a lot of setup, a lot of having standards, susceptible and resistance. And we try to build on that and continue and actually do better if we can. Just a bit of, it's not about me, but I thought it would be nice to tell you a bit of the work that I've done. It's all coming together, ties the room together, like the Big Lebowski rag. Anyway, this is work that I did 15 years ago. We heard that there was a new herbicide coming to Australia, and that would have been Sakura, commercialized in 2012, but a farmer from Wongan Hill, not far from here, that sat on the AHRI board at that time, asked to Steve, well, why don't we try to see whether this herbicide can be broken? And so Steve came to me, gave me this idea, and I took off with this population that was the first one to be identified to be trifluralin resistance in Australia in 1982 in a paddock over east, and I selected with low rate of Sakura, and I could select Sakura in only 3 years. And again, this was really the needle in the haystack, because Sakura still today, it's at the very least in WA, is very rare. So, but that population had the trait, and that population really provided a new perspective that there is no new herbicide that is, as we know, bulletproof or can be broken if it is not used properly. And so all of that work and led to other work, and led to, with Peter for many years of good extension, and try to understand that how these herbicides can be detoxified, or how this cross-resistant patterns, very difficult to interpret and put in one single box, can be understood better. This was the same population with, exposed 3 years to Sakura, it was resistant to Boxer Gold. So again, it was a very big deal at that time, because Boxer Gold and Sakura were the 2 new things. There were not too many herbicides, there was no Luximax, there was no Overwatch yet. So again, 2 brand new herbicides almost just released, and here you have the proof of concept that shows that cross resistance can extend to the 2 of them if you only just use 1 of them. And that led, again, with the help of Peter, to try to simplify. I mean, resistance is very complex, and if you try to describe all the very details, we get lost. So we try to simplify with a simple sort of diagram, and we put in different boxes what we believe and what the science behind was suggesting that we should stay in different boxes. Meaning, if you use trifluralin, you're not going to get Sakura resistance, and if you use Sakura, you're not going to get trifluralin resistance. So that has been now defined at the very fundamental level. All these letters are referring to the mechanism, to the DNA, to the protein that allows these ryegrass to survive. And so they sit in different boxes because they are different. And they are all different to propyzamide. You can use as much propyzamide as you want. You won't get resistance in ryegrass because it's very rare, and it has never been reported apart from a minor level, which is quite doubtful sort of set of data. And so, yeah, we simplify that, and we put in the same box Boxer Gold, Avadex, and Sakura. So they used to belong to different groups. Now with the new system, they all belong to the group 15, but they used to be J and K, and still are J and K when we want to refer to something that we learned with so many years. And that was you know, kind of contradictory, because they belong to different groups. We learned that, you know, we should use different, rotate the groups, not just the herbicide names, but we put them in the same box because we believe, and we had some data that was suggesting that the rotation between Boxer Gold and Sakura would not be as effective as the Trifluralin Sakura type of rotation. But once I did all this work, then I learned that in actual fact farmers most times use mixtures. And so I didn't have any data on mixtures. And this is just a photo that tells more than 1,000 words said with an Italian accent. That you know, this is what I saw in pots.
We just need to explain that each one of these cells is a different paddock. Is that
correct?
Yeah,
this doesn't need explanation. You don't need to be a scientist to see that box over there is very different from the two here. And so
but that's 20 paddocks, right?
Yeah. Peter is obsessed with details. You know? You see, I am at the extension here. He's the scientist.
Oh, what I'm getting at is you could say, well, that's one paddock, and yeah, you've just picked a good one to take a photo of, but that's 20 paddocks, and so that's pretty representative. That's my point.
No, because we are still in the introduction. Just
sorry,
imagine if we go.
You told me to interrupt. You know.
Satellite and micro.
Definitely, each of these, when you submit your samples, the samples get processed and prepared, and then we count manually. Not true. We count with a machine 100 seeds, and then we put these 100 seeds into this little 5x5 square, and that is one paddock, and that is one herbicide. This is 4x5, 20. So 20 paddocks in. So if we can put all your farm into one of these little trays, and then we spray 30 different things. So we have 30 different trays, and we represent all your farm, millions of hectares in one single tray. And so yes, each of them is very different. And so what I saw here is that there was a bit of trifluralin resistance there, and there was a bit of Prosulfocarb arcade resistance there. But when we put them together at the same rate, 1.5 liters and 3 liters, everything was almost controlled. This photo was taken like 3 months after, you know, the herbicide had been sprayed in late April. And so of course there was something that emerged because maybe it was a dormant seed. Dylan.
Roberto, we see with, like, particularly with broadleaf, if I've got any sort of resistance, like we've seen ester resistance translates to like antagonism when we're using it in a mixture. Do you see any resistance traits with ryegrass that antagonises as a mixture as well, or are they all pretty much, we've got a little bit of resistance, just we keep adding other things?
Yeah, it's case by case. I mean, the most famous one is 2,4-D and diclofop, but I don't think anybody would rely on that for ryegrass control. That was very clear that the 2,4-D basically made the plant tolerant to diclofop. So you couldn't just control with one pass dicots and monocots because the 2,4-D was antagonizing. There are examples, and I will go through one when we talk about radish, about the MCPA and velocity. That's something that I found this summer. In terms of Pre-em, I haven't seen any antagonism, apart from there was the odd result that I could kill a ryegrass plant with Triflan, and I could kill it with Sakura, and then I used the mix, and there was some plants surviving, but that was not the norm, was the outlier. Because in the tank a lot goes on, you know, there has been discussion over and over and over whether the group Gs antagonize a bit glyphosate. I did a bit of work, and I think that eventually there is always they always add. Now, I mean, if you use a group G with Paraquat or glyphosate, it gets very technical. So yeah, sometimes you get something extra than just 1+1, and sometimes you get something slightly less, but none of those mixes lead to failure where you have full control with one or the other, you put them together and it doesn't work. No, that doesn't happen. In fact, this is most likely what you get as an outcome. You get more, and it's logical.
Are we allowed to call that synergy?
Well, you are allowed to call that synergy if you do the analysis, and you do the Colby analysis, and then you do the stats, and if the stats tells you, then maybe. But still, synergy is very rare. Synergy is velocity, you know, group C and a group H. That is very rare. Sometimes you can get the same with group F and group H. You know, they are two bleachers and they do kind of deliver more than additive. We get into radish country. But yeah, it's very definite. Sometimes, yeah, the synergy is something that, the sum of the two is greater than, I mean, the result of putting two together is slightly greater, or significantly greater, statistically greater. Yeah, we will never get to the bottom of this presentation, I'm telling you now. So this is the bigger picture. We are still in the introduction only. You know, we want to grow more crops and we want to control weeds in a very selective way. I put this not by chance because this is the new guide that came this year, Overwatch, but this is what we want. Full selective control, preserving the crop. You know, this is what we get in pots. We spray ryegrass and, you know, dose response. This is survival or biomass, whatever. You know, you hit it at the field rate and you get really good control, 95 or greater, and wheat is kind of immune. But when you get resistance, this line comes back up here and you don't get this separation, and therefore your wheat control is very difficult. I will present the study that I did. I only presenting the data from this GRDC. Of course, as Peter anticipated, the study was larger, and I will just give reference to the larger study from last year or this year just to provide some context. But this year, 30 herbicides times 100 population, so that is 3,000, and 100 seeds per sample, so it's about 300,000 seeds that tested. So the density of this data is quite high. It's not just one sample or one population. And I will present in this order, which is probably the order that you follow when you spray, with the knockdown first, pre-em, and post-emergence at the end. And yeah, this is how, when you submit a sample, this is the sort of arbitrary, this is not defined by any measure. I decide to call something that is 5% or lower survival susceptible or developing resistance. This is the category where everything happens. This is the most important one, and it is very difficult to define because maybe you haven't seen yet in the paddock, but if something is brewing in the background and you need to know. And sometimes it doesn't match what you have seen in the field because of the conditions are always difficult. I spray plants in mid-April and you spray maybe, you know, mid-May. Very different rainfall. I water every day, you get whatever rainfall you get to activate your pre-ems. So, and we will discuss about those differences. If the survival in pots is greater than 20%, sure enough, your perception is matching, and Fiona will discuss about that, is matching what we see in pots, and it's just a confirmation. So we start with the good news, with the knockdown. There is not much to say. Everything was susceptible to Paraquat. Sure enough, this was like a photo that I've taken after the second spray because I had some plants that emerged later, and sure, you can still see that no matter how many times you apply, there is always a late emerging ryegrass. Oh yeah. It's not good. I don't even know how to use it. So yeah, there is no resistance to Paraquat. I haven't seen it in the couple of years, so actually 3 years of resistance testing that I've done. I know that there has been some anecdotal evidence that there is some Paraquat resistance that has evolved in broadacre situation. I've never seen. And so the only anomaly is the plant at the back there, which is ryegrass that comes from South Africa. Has been selected after 20 or more years of using glyphosate and Paraquat, and so this one is resistant to glyphosate because of a mutation. It is very resistant to Paraquat. You can pour gramoxone on it and it's not going to die, and that, unfortunately or fortunately, that is my standard. Never ever I've seen anything that looks like that. you always have this, you know, half a leaf that is half bleached, and the plant is very suppressed. So it's very difficult for me to call that resistant. Sometimes I would report one surviving plant out of so many tested if you submit the samples in a resistant testing, but yeah, nothing has ever looked like that. And if you respray whatever is a shy survivor to the first pass, nothing survives. So in my opinion, there is no Paraquat resistance. I don't know what your experience is with Paraquat, whether it works or it doesn't work, or yeah, in the field may be different. Anything to?
I'll do a little, you do this one and then we'll cover knockdown one.
Okay, cool.
All right.
So yeah, we move into glyphosate. Yeah, glyphosate was different. The situation this year, 13% overall of these, and again, for the GRDC, I only tested about 100 samples. So 13 of them show some level of resistance, 9% developing, so this 19% survival, 4% highly resistant, and they look like that. And I sprayed yeah, 1.5 liters of Crucial, which is the triple salt product from New Farm, so 900 of active ingredient. Very good rate to discriminate what is susceptible and what is not, and also picking up this evolving type of populations. So very confident of this data here. We don't go back, we'll go forward. What was different was that there was a significant increase compared to, in WA, there was only 3% in 2020. In 2020, I used Power Max at 2 liters, so there was a slight difference in the rate delivered, 180 of active. I don't think that 180 justifies, the much lower frequency found in 2020. When we gave this workshop, around the state, there was a lot of feedback that they had, maybe this year was the first year where the knockdown was used, and they saw way more plants surviving than we would have expected. So probably the 13% is scary because it's a significant increase. I'm not sure yet whether this is a beginning of a trend. We don't want that for sure, but yeah, it was the data that we got. And I can stop here
instead of pause there for a second. Yeah.
Instead of going to this next slide.
Well, it's on there now. We better talk about it, otherwise we'll wonder what it is. So,
well, I mean, it's just that as we mentioned already, that in the tank there is a lot with glyphosate. There are a lot of products that provide some spike, like Group Gs or Overwatch. There is a lot of talk that is actually providing good spike. Or sometimes in a Roundup Ready canola you can put Clethodim. Clethodim is definitely a ryegrass herbicide, and yeah, I've tried the mix, and I can tell you what the number is. Actually, I can tell you what the number is when you mix glyphosate and you have glyphosate use 13%. What do you think the resistance will be when you use it with Clethodim 500 ml?
What's the Clethodim resistance?
The Clethodim resistance, I anticipate, it was 34% across the state. So 34 and 13. What do you get when you use the mix?
2.
5, 2.
1.
1 hit.
I said 5.
5. Yeah, very close. I mean, it's the percent was 3. So almost the product of the two, which makes sense because they are the resistance is provided by a major single trait. So you have glyphosate or Clethodim, very different and very difficult to combine the two of them. Yep. And I think that sometimes there was on Twitter. We all use Twitter, unfortunately, but there is a lot of discussion going on, and it seemed to be that those two can't stay together. Maybe the system doesn't allow them to coexist because very rarely you spray glyphosate and Clethodim in the same season. I don't know, but yeah, it seems that the glyphosate that was resistant to glyphosate was not to Clethodim. So I was quite happy with the result. I put it on Twitter. I wanted to be famous, and I got hammered because no way, you know, you have to. Actually, my intention was to specify that it would have been a good use on a Roundup Ready canola, and yeah, over East they killed me because no, do not suggest that this should be a knockdown strategy because it only can be as a crop together with crop competitions. But in a Roundup Ready canola seems to be a quite valid type of solution.
All right, let's just spend 5 minutes talking about knockdowns. So Roberto did this study. The AHRI Insight will be out probably early next week, and showing the 500 results from the whole country. And in New South Wales, 42% of the paddocks had glyphosate resistance. Right, it was a bit of an area where it was sort of suspected, but glyphosate resistance is a big problem in New South Wales, Victoria, SA. Speaking to a Victorian agro earlier this year, he said he witnessed the fastest adoption of a new practice in his 30+ year career. This is Phil Hawker, who used to work with the elders, and that very fast adoption was going from the glyphosate Paraquat double knock to double Paraquat. So all of these growers in Victoria that had glyphosate resistance very quickly have moved to double Paraquat. So this is just a little look into the future. So with our sort of current glyphosate resistance levels in WA, I think the glyphosate Paraquat double knock is still probably best practice, and there's a whole suite of other weeds. If you ever get a double knock, that is. I mean, you guys probably dream of a double knock. But I just wanted to sort of peer into the future a little bit. So if we go Paraquat, so we've normally had our glyphosate followed by Paraquat. And what do we put with these ones? Do we have a group G with this one, with the glyphosate, or with the Paraquat, if you're using a group G spike?
Yeah, historically no, but now generally yes.
With the glyphosate or the Paraquat?
Split.
Bit of both?
Half right by the time.
All right. Could be either. And where's our pre-em go? Pre-em goes here, right? So one of the big messages from, and I don't know if Roberto will back this up, is to not put the group G with the glyphosate, because group G is, well, let's use the correct terminology, group 14. And we're moving to the numbers now, aren't we? Group Gs, group 14s. One of the sort of talk amongst researchers and so on is to not put it with the glyphosate, because we can antagonise the glyphosate a little, and it's better with the Paraquat. Roberto?
Well, I mean, again, there is antagonism between the G and the glyphosate, but that is very different from failure. So it's very difficult because if you do the experiments, you see that the G adds. Okay, apart from the speed of burn out, it adds something. The problem is that when I did some work with highly glyphosate resistant ryegrass, and you realize that in that scenario, whatever level of control you get is only the G that provides that, which is never good enough, because otherwise, you know, like the group Gs, the Terrad'or, the Voraxor would be registered as standalone for ryegrass control. And so it's a very difficult thing, and in my opinion, it becomes an opinion. It's not data driven that, you know, to use it together it's a good thing because it adds, and so you can't deny that. But yeah, it is slightly less than what you would expect if you do all the maths and stats.
Okay, so yeah,
Roger. The old group, it seems to be like group G is such a like the old group G's, like when we had our failures with fly, was like the old group G seemed to be a lot faster, like Ecopar and Hammer and Nail, where you didn't have GPS, you could see where you've already been in the paddock because they work that fast. Where the new ones seem to be maybe a little bit less riskier, or like, you know, not like a bit slower, which I think might, like, but I know we've been once burnt, like twice shy sort of thing.
Yeah.
So I don't know.
Yeah. Roger, you got another comment?
One of the comments around what we did this year, the split application they're talking about, is there was a lot of green material out in the first knock. If you didn't get a fast burn down, get that stuff burnt off, you had green tissue there, your pre-em didn't make it to the ground, and you had big patches that it's almost like you hadn't sprayed, it just hit green material.
Yeah.
And Sakura hitting green material doesn't work. Luximax hitting green material doesn't work. it needs to hit ground. So the fast burn down, so the split application, Falls developed it for Voraxor. It seems like it's working gold. 100 mils, 140 mils, best of both worlds.
Yeah. All right, we won't dwell on it too much. I know in New South Wales they're sort of not doing too much of that because they've got quite a bit of glyphosate resistance. A good robust rate of glyphosate will sort of knock the ryegrass around because glyphosate resistance in ryegrass isn't generally really high level, is it? It's sort of 4 to 8 fold sort of thing. So yeah, good conditions, really good glyphosate. They're avoiding it. So not being prescriptive here, saying never do that. I'm just sort of relaying what's happening in these areas where they've got a lot of glyphosate resistance, right? And then if we talk about this Paraquat followed by Paraquat, right? What's going to happen there if, you know, Victorians switch from, you know, blow up glyphosate and then switch to this? What, you know, what's going to happen? So how do we look after Paraquat in this situation? And so it is about these mixing partners. It's about having the Group G and the Pre-em with them. And you know, we've got some Group Gs that might be giving 50% standalone control. Is that fair enough?
Yeah.
Yeah, 50%.
Yeah,
So
and the Pre-em is a big deal because there are Pre-ems that have no post activity like Trifluralin and Pre-ems that do, like Overwatch.
And even if it's not post, you know, you might have a plant up and it might survive the Paraquat, and then you get some rain washing it into the roots and it can kill it later, can't it? So sure, we are concerned about double Paraquat, but if we wanted to work out how to look after herbicides in these high pressure situations, it's about what we put with it.
Yeah.
We're right to leave that one there. Is that all right, Roberto? Any other questions about knockdowns before we move on to the next
How
week?
far apart Paraquat?
About a week. Yeah. And so what Roger was talking about there, like you got green paddocks and you want your Luximax to work or whatever, Paraquat's a good one, you know. And yeah, I mean, in the past we would have just been shouted down suggesting things like this, and no, you've got to use different knockdowns and so on, and you do, but I mean, if you've got glyphosate resistant ryegrass, what's the point of putting glyphosate on it and then following it with Paraquat? Like, it's just doesn't make sense, does it? So in the ideal world, we do the glyphosate Paraquat for years and years in a row before we have any resistance, but we know that there's all sorts of seasonal conditions that just don't allow that. What Roberto's talking about here is another good example of it. And, you know, I think it's a great example of if glyphosate's a big threat in your farming business and Roundup Ready canola is an important part of your business, that's good strategy, I think. Same thing, mixes.
Before we move on to knockdowns, glyphosate glyphosate, what is that heading knockdown and in crop with Liberty coming?
Yeah, just fill me in. What's Liberty coming in first?
Or canola, so it'll be applied glyphosate.
Yeah, that's right. Yeah.
Glyphosate.
Yeah. Well, Roger will probably comment. I think you probably know more than me. But my understanding so far is that Liberty really needs high humidity and is it warmer temperatures and high humidity? Is that correct?
Yeah.
Yeah.
And 2 sprays.
And 2 sprays. Yeah. And so I guess we all have our reservations as to whether or not we'll get Liberty, glyphosate, or used to be called Basta, working in this environment. I don't know if we've got any advances on that. Have we got any?
It worked great this year.
Yeah. I certainly did trials with it back in the day, and it, I know didn't get any good results out of it, and it was warm and dry, so it was too dry. And I heard the story that in Canada, when they first rolled out Liberty Link canola, and someone else might be able to confirm this, Hugh, that farmers sprayed it, and whenever they had a failure, the farmer called them and said, we've got a problem here, we've had a failure, and they said, right, what conditions did you spray it in? Okay, what you need to do is this humidity and this temperature, here's some more Liberty, go and spray it, and they had good results in general. Is that sort of somewhere near the story here?
Yeah, although in my experience, in Western Canada, it's characteristically low humidity and cool temperatures during spring season, and usually Liberty worked very well. So,
yeah, okay.
So that did happen, but not very frequent.
And
Can I
so,
just ask, Peter, Hugh White, was by far the most, I've been through there, by far the most predominant, like, the system. So how did that take lead over the glyphosate?
Genetics, the yield.
Yes, so related.
Hybrids, yeah. And now that Liberty's off patent, cost has come down, so it's really a jump on the techno.
And Peter, could I just add to Roberto, in theory, I mean, the double knock is just something we dream about, and it's just fantastic, and this year we had the opportunity to do it. But I think we've just lost so much ground through a lot of WA in the last number of seasons, particularly up this way, as you're well aware of. We have to go back to 2016 before we had a classic double knock year, and so we've had 17, 2017, 2018, 2019, 2020, 4 years where we virtually didn't even have a chance for knockdown, let alone a double knock. And the crops went in, by and large, farmers farming up here know that the crops pretty well, almost the complete program went in dry. It only rained about the last day or two of the program, or even a couple of days after the program was completed. And I think it's been the most consecutive years that I can ever remember farming, that we just had no knockdown opportunity. So we're just throwing all our reliance on the in-crop herbicides, and then having sketchy seasons then for even those to be activated, so often soil moisture activated. So it's been a very difficult time, and I think contributed to a hell of a lot of the problems we've got, in terms of work
just
being
no
able to get
knockdown,
through.
yeah.
Just, it's really good science, but we just haven't been able to employ it.
also, farmers have increased in scale, so you just have to get seeding done by a certain time. We know all the benefits of early seeding, all of that, and a lot of what we've talked about in the past is just having a low seed bank just enables that kind of farming, doesn't it? If you've got a high seed bank, you just can't put the whole thing in dry and hope to have a clean farm, can you? So it's all related. So we'll keep addressing that, I think, as we go with the Pre-ems. I've got one last thing to say about knockdowns, and that is, I think for up here, the big risk of glyphosate resistance is from fence lines. So we're just going to destroy our best herbicide for the sake of a fence line. So I just think that's got to be another area. We're not going to spend time talking about it now, but that's got to be, I think, the focus of preserving our knockdown there.
Well, I mean, very frequently when I hit a resistance sample that comes from a fence line, glyphosate resistance sample, very frequently. That is almost the norm. So yes, Pre-ems, the method, as we touched on that, each little square has 100 seeds, and they do get directly sprayed with the herbicide, and then after half an hour or so, I cover with half a centimeter of potting mix. For this experiment, I used a potting mix that was less with less organic matter and more sand, just to get closer to what type of soil you might have. Still, the level of organic matter would have ranged between 3 and 4%, which is much higher than what you have down in Albany. They were happy that it was quite close to what they get. And these are the data. I did 3 doses of Trifluralin. If you have received the test, you would have seen I found that is very good to, within the single test, do different doses, because we do repeat the same thing over and over at different. Of course, we can't just do the same things in reps. This has to be done quickly and to a certain scale. So I decided to use a liter and a half, and 3 liters. Pretty much going from 1.5 to 3 liters. The situation didn't change much in terms of trifluralin resistance. And what I report here is the actual value of what I counted in pots, assuming that the seed germination would have been 100%. This is not this is the actual percent survival, so it can't be lower than this. This is the best case scenario of resistance. You will find it slightly lower than what you might have in your mind. You might think that trifluralin resistance in WA is like about 20 or 25 or 30%, and here is 13. Well, first of all, the sample, the sample size wasn't big. It's only 100 samples, so it was only what I got. In some areas there is no trifluralin resistance, and trifluralin is still delivering an amazing good job. You see plants surviving, yeah, good. But in each of them there are 20 samples, so you know, overall this gives you a very good impression that in some 20 paddies there is none to a liter and a half. But that was the soil I used, and that is, you know, if you see higher than the tray, that is because I cover with a certain amount of potting mix that holds some water, and then we frequently water. So yeah, that was the figure of trifluralin. Moving on Sakura, I used the new formulation, the Flow. Yeah, you can see that there are plants there because I had some samples from South Australia and Victoria within this common thing, but in WA, Sakura resistance is very low. 1% is as low as it can get. When this data will be analyzed and elaborated and expressed as a percent of the untreated control, which is usually what we do, that level can rise maybe to 5 or 6%. Well, I mean, this is the business model is to produce a paper like this every year with all the testing that we do, because if this data get old very quickly, next year we want to know a bit more, not just because we can't remember, just because we think that the season has been such and such, and you know, I couldn't desiccate and I couldn't do this and that, and so the weeds got away. So we need every year a few numbers to get, you know, to keep at the back of the mind, and so this is why we started the testing service, and this is what we want to do every year, give a summary of what we've seen, and in terms of Sakura resistance, it's very different around the country. This is what the results are from last year. So you can see that the situation in WA is about 8% when we express the data and we clean the outliers out. So it's a marginal problem. There was only a couple of populations where really there was a high level of survival to Sakura, the full dose in 2020. In South Australia and Victoria, the situation is very different. You know, the level of Sakura resistance is very high, almost 50%. And you can see even it's not just more spread geographically. Each sample survived to a greater level. So I don't know whether that is, well, I don't know, I should know, but it's probably because they had more use of Sakura on much more difficult to kill trifluralin or trialate resistant population. They had used quite a bit of trialate in the past, and I've presented before the unique box of all them together J and K. Now it's all 15, so it's easy. We get used to these new numbers. But yeah, the good thing, we talk about mixtures already, and yeah, I just simply mixed trifluralin 1.5 liters and Sakura, and yeah, the problem seemed to temporarily disappear. You know, very, very, very clean. There was no, there was probably a single population from South Australia that had the two together. So zero resistance to the trifluralin Sakura mix. Switching to Luximax, within this project, zero problems. Roger is there, and so yeah, there was no resistance in this in the sample submitted in the GRDC. You can see at the background here, I put a very blurry so that you can't see them. There are some populations that survived the full rate. I also did 3 doses of Luximax. I went off-label at 600 and 750 mils. They did survive. I went low-level, and they did survive. And I did the dose response, and they do survive. So they have. But when we have received from BSF C14 label radioactive Luximax, which I tried, you know, a couple of drops just to see the radioactive buzz, and it's pretty good taste. But in the lab, so we can detect Luximax in the lab, and Danica Goggin is working on this. You know, she's the best. Together with Hugh Becky, she's the best researcher we have in AHRI. I'm just a technician, I'm just nobody. But yeah, she can't see, the level of resistance is very low. These populations have a very, at the very best, a two-fold type of resistance to Luximax, and we are working on the hypothesis of, detoxification, metabolism. So it's good news. We all know the Achilles heel of Luximax. It can can have some crop effect, but wheat can detoxify Luximax better than ryegrass. And ryegrass, the few populations that I have identified, these are the biggest one that I've ever seen. They are still low, it is still low level. So this herbicide is robust in terms of resistance. And then I went full BSF mode, and I did try Luximax and Voraxor, and BSF funded the service stop. Huge now. Now there is no conflict of interest. Of course, this service would not exist if BSF last year didn't seed fund, because they gave me the confidence that I had some money to pay the salary of technicians doing all the work. I'm only here to talk, but lots of work is in the background. And again, this was really no plant surviving. Even the few that were somehow able to survive the full dose of Luximax with the Voraxor were killed. So yeah, no resistance at all, and so it was quite surprising, because we do not understand the interaction between Group G and Group T, or whatever Luximax is, Group 30. But yeah, there is something very interesting going there. Of course, we know that Voraxor has also soil activity and pre-emergent activity on ryegrass, but it was really interesting. Different situation with Prosulfocarb. The photo looks terrible because it looks like full of ryegrass. Well, in this potting mix with 3 liters of arcade, even my susceptible survived, and this was what the susceptible ryegrass looked. So I had like a 1% type of response, 1% survival, but there were a few of these samples that had many more. So I could understand which one was resistant and which one was susceptible. And overall, even though the photo is not great, what I could see was only 5% of samples that have a level of resistance to Group J. I didn't, this year I didn't test for trialate. I did last year, and usually the levels are very similar. I keep going if you don't stop me. I did test Ultro. Ultro will come. It's not available yet. It's a new herbicide from Adama. It's carbetamide. We are still learning how to use it. I mean, I use it. There is a range of doses that you can use. I sit on a dose that would have been $35 an hectare, so 1.1 kilos. The work I did 2 years ago, I used at 1.5, and no plants survived. So I knew that I used a bit lower, and overall about 2% did survive to the point that I assessed as resistant. But we are working on it. Of course, when we have a resistant sample to a new herbicide or refurbished to be new, we then keep those surviving plants, and then we bulk them up, and then we do those response to confirm what we have seen.
Roberto, with the herbicide, like, because Ultro, obviously we don't really know is using Ultro yet. Why and how is that included in the mix? Like, is that funded through, is it Adama?
Yeah.
Yep. That fund that to be included across the board, or do we add that in because we're just interested about what future herbicides, and is that funded through everyone to include that that testing, because we're worried about like future herbicides?
Well, I did use it because I wanted to. I have the genuine interest in knowing whether there was some resistance to it. I read a paper written by Chris Preston at the Crop Updates in Adelaide that he was suggesting that there was some low level of resistance in ryegrass that he collected over there. Yep. I just wanted to check. Yeah, all this 30 herbicide option is something that I decided. I made a decision based on feedback that everybody can submit, and suggestions from agronomists, and genuine interest, and then yeah, I came up with a list and I put on Twitter, and I received some more comments, and then I changed it a bit, and so eventually I solidified, and then I started the experiment that lasted, you know, I was only, we had 500 samples, so every day we would plant 2 sets of samples, 50 trays, and then spray 2 herbicides every day. And so by the time I start to, you know, to go through all this, it's 15 working days, so it's 3 weeks, lockdowns or whatnot. And I see that some of the herbicide didn't work as well, so then I came back with, you know, like Overwatch followed by Clethodim, and also I started to add all of these mixes, or try to fit a particular system, thinking about canola. So you could have come back with Clethodim, and so the results that I sent back eventually, they have all these options listed. Yeah, this one I thought it would have been available. I didn't actually know that it wouldn't. It would only be next year. But yesterday there was a tweet from Barry Haskins that he was comparing on his farm or his research sort of field. And yeah, it was comparing with Trifluralin, so they use it together with Turbine, and yeah, of course, it was showing how good the Ultro is compared to. It's only for lupins, it's not for canola. It is selected in canola, but they couldn't get registered.
So Roberto, sorry, on that one, do we think that that is likely to develop resistance quickly in the field if there's already some trend towards it here?
Well, it's only 2%. What I wasn't, I mean, I wasn't happy with the way that the plants responded, because it seemed that I had survivors always more consistently on the edges of the trays. So I think that I started to exclude many of them, but then, you know, in some cases, I would be quite confident this is just not by chance. Again, this is only one rep. We always have to apply the scientific method. Whatever we see, we are, yeah, good, very good. Now you have to repeat it. So we are in this phase. This is our sort of preliminary data, good enough to, you know, tell you exactly what I found, what I've seen. We are still working on it. 2% is very low, and it is rate dependent. So I'm not sure what rate will be used. Maybe the rate that, you know, farmers will use it in the field, first of all, will be used, maybe mixed with something else that may, you know, the metribuzin or whatever, that may bring some additional level of control. So, yeah, as a baseline on a plain field where everything was sort of tested within a couple of weeks, this is what it is. So you compare it with your 0 Sakura or 13% of Trifluralin. So you have a 2. It's not a 0. If it was a 0, I would have found a 0.
Do you think that changing the body media would change the results, Roberto? Like going to like a more loam soil or a clay soil?
Yeah, there are some herbicides that are more sensitive to the organic matter. Trifluralin is not. Trifluralin is not in summer or winter, is exactly the same. Sakura is sort of the same way. Maybe solubility, I don't know. Some other herbicide, Prosulfocarb, doesn't like the carbon, and I'm still working to find a system that is unique. And again, it becomes philosophical, because I don't want to report false positive. On the other hand, I want to see things. I don't want to overlook 2 issues that are underlying the field. Last year I got criticized because I didn't find enough glyphosate resistance. It was only 6%. And then I retested, and yeah, in winter things changed a bit, and then eventually I repeated again with ammonium sulfate, and eventually I had a figure that allowed me to be confident enough that what I see was as close to accuracy. We are always shooting in the dark, or relying on sometimes something that is quite subjective. So yes, there are some herbicides that are sensitive to the organic matter of the potting mix. One is Prosulfocarb, the other one is Overwatch, that I'm going to talk and show some of the data. I don't think that do everything in river sand, wash river sand would be the way to go, because everything would be probably susceptible. And in the field you never have that situation. You always have the green material that stops some. So you always are, and actually, I'm using robust rates, and Hugh can back me up on this. We got criticized by this guy in UK. You know, he told me, no, you are working for a company. You know, a company will use your material too. I say, well, I don't care. Seriously, I stick to my philosophical line, which is I hate to report false positive, and I try to close the gap between pots and in the field. In the field, you don't use a cut rate. In the field, you use a field as robust as you can. You know, that makes economic sense. And I have to use the same, otherwise I'm already disconnecting from the real world. Yeah, it's always sort of try to find a compromise that, you know, fits, one size that fits as much as possible.
So could it be possible that those plants that have got survivors on the pot walls, it's leaching the chemical out of the soil there, like it's running down the plastic and taking it out of the root zone?
It could be. What I found is that the girl that was watering the pot, she was really keen to water these pots, and she was holding the hose very close to the trays, and sometimes shaving off the cover. So again, we repeated those many samples that had this problem, and then we did a sort of average of the two. But yeah, it's just good. It's good that it's not used the herbicide. So it's definitely some background information and experience that we will use going forward if we test it again. So then those issues don't repeat. But sometimes, you know, again, new herbicides, we don't have a resistant control yet, so we don't know what the resistant population will behave, how it will perform in that same environment, and so it's also difficult for that. And so we only have susceptible ryegrass, ryegrass that we can kill, and if that is killed, then whatever survives is difficult to ignore within the limitations, provided that we are not doing a systematic error. Then if I kill the susceptible, I can't ignore, otherwise, you know, what's the point of doing this? But yeah, we try to optimize as much as we can. I'm really confident that 2% is not a false positive. It's really something that, you know, shows a level of resistance.
So just on that low rate idea, I think that when we got those results, I think those low rate, like those low rates, especially the pre-ems, were really interesting, because in the field, when we have a time system, we get, you know, the in between the row against the full rate, then we always have where our problems there are in that shoulder, and it's either getting 0 or a low rate. And so that's why I think when I look at those results, I think having those half rates in there is actually really interesting to look at, because I think it helps connect to that problem on the field. So not that it suggests that we should go to half rate or anything like that, but yeah, just...
No, well, I mean, the point that was made to me by this researcher is a legend in UK. He's like equivalent to Steve Powers, so like very big ego as well. And he thought that he could have made an impression on me. I told him, look, we are 10,000 k's distance. Don't care. But I did care, because in the end, science only progresses where if you get criticized. If you get criticized, you really are forced to go backwards and check again that whatever system you are developing is good. And so I took on board, and he was telling me, look, work on rates that give you maybe 80% type of control, because those are probably more likely to reflect what the farmer would see in the field. You have 80% efficacy, which could be still acceptable in some situations. And so he had a very good point. And that's why, yeah, the low rate, sometimes we see those results that are not realistic. We can't expect that 50% of the samples are resistant to Overwatch. You know, that doesn't reflect what has happened this year, definitely not. But it's good to have it there, provided that we are prepared to take on board some extra data and not just be scared of, but, you know, having some number there that maybe in the future will be useful, or immediately useful to warn you that, yeah, if you do cut the rate, maybe that is the worst. In a worst case scenario, in a situation where your herbicide is not activated, this is where you end up being.
Get you on for 5 minutes, mate, and then we'll have a big pre-em.
Well, then we have a break because Fiona wants to talk. She came all the way from Minginu. So this is Overwatch. Yeah, 8% of the samples were classified as resistant. We mentioned already that, especially at the low rate, even the susceptible got knocked and then recovered, turned green again. I knew that that would have happened because I worked with Overwatch in the last couple of years, and I know that this may not reflect the amazing efficacy that has been seen across the country sometimes in particularly highly activated conditions. But I also have some data that show that it's relatively easy to select for resistance to Overwatch, and there is already data that has been peer-reviewed and published that shows that some ryegrass populations resistant to group J herbicides, when they were in a control environment selected 2 years in a row with Overwatch, all of them had resistance. So in my opinion, this 8% is not inaccurate. Is quite truthful, and I try to be as conservative as possible. But some populations were just, you know, even beyond above the label, they were surviving and recovering. So just a few photos of what I've seen. You know, you want to see a lot of pink material. Overwatch doesn't stop germination. The plants germinate and then they die. And it's very easy to see. This is below label, by the way. It's very easy to see where you have green ryegrass and not just the usual pink that you would expect. So, yeah, this is what I've seen. This is the same, but above label, just taken, you know, just a bit after, and there were a few of those populations that remained green. This is what I had seen last year with a different potting mix, and again, sure, there were some pink ryegrass, but overall, this shows you that you know, ryegrass is already fully loaded of some traits that allow detoxification. And so, yeah, I saw this, and then last year I did the work trying to go from 0 organic matter, and everything was pink. You know, I had each row is a different population. I had like 4 different reps, and at the back there I had a population that I selected and now show some level of resistance. So, but I wouldn't see anything when I use river sand. This is the field rate. And then I use Gin low, and I start to see some ryegrass at the back there that is green, while, you know, the susceptible control here remains purple. That's fine. And then when I use the potting mix, you know, sure enough, now the ryegrass there maybe is just tying up some herbicide, and those plants look very different, and they are able to survive, and also those ones at the front, some of them survive. So again, we are still working on it. I've just started this week, went back, and I will re-characterize with dose response up to 3 times the label, cutting down to a quarter, and characterize what I've seen in 2020 and 2021. My deadline is crop updates this year, where I will have a bit of a much better idea of how widespread the problem is and how big the resistance is to this new herbicide. You know, this year I grew my first crop. I did my first field trial. It's good after 16 years working on herbicide resistance, and so I had a small GRDC funded field trial in collaboration with the Uni of Adelaide in Shenton Park. Very sandy soil, sown on 2nd of June. AHRI is on wheat, AHRI is the variety, Sakura, I mean, we introduced the ryegrass, so the seed bank is not very big. We hope that some ryegrass will survive, because the ryegrass was highly characterized in 2020 and then properly mixed and delivered. And I actually also introduced some of the population that were tested in April survived Luximax, because Luximax was also part of the experimental design. I tested Voraxor and Trifluralin, Sakura plus Trifluralin, Sakura plus Trialate, Luximax, Overwatch, and untreated control. So yeah, what I wanted to say is that I learned pretty quickly that in a sandy soil that has been cultivated several times, trifluralin is very damaging. Number of plants, 160 per square meter with Sakura. When I used Trifluralin, yeah, the mix works very well, but it was very damaging. Overwatch, as good as Sakura, if not better. The best one was the untreated control. Well, again, yeah, we talked before about herbicide selectivity, and so we need to be very aware that in a perfect world we wouldn't use any herbicide. We are forced to use them in a selective way, IBS, whatever. Overwatch, FMC told me that they had never seen the level of bleaching that I've seen in Shenton Park. It was really bleached for 5 weeks, but now is the best herbicide. So it's a continuous sort of compromise. Yes, I've seen some resistance in pots. I put in the field something that I believe is like 1-2%, and Overwatch has just killed everything. Yeah, so always learning, and I try to put myself in the most difficult position and try to learn as much as I can. Just the data to finish off, I mix it with Trifluralin, and again the level of resistance decreased. But what I saw frequently is that even with the mix, you know, Sakura and Trifluralin went to 0. With Overwatch, at least in pots, with Trifluralin I couldn't go to 0, and this was probably the best treatment. I put it with Sakura. Yeah, within this project resistance went to 0 because Sakura worked really well. But yeah, it was weird because I couldn't see the plants as bleached as when I use it with Trifluralin. So again, we are doing all this fundamental work in the background, and we are still learning how these herbicides do work. And so, of course, I haven't addressed the other issue, but I'm working also on this, and I have to say that you know, FMC has been really supportive. I just wanted to educate myself, and I did a little experiment to see whether it is volatile or not. It is classified as moderately volatile according to the GRDC tables, and I think that value is actually true. I wanted to a proof of concept to make it happen, so the looping wasn't sprayed, but I sprayed all those pots around it. And if you ask about the area of those pots, you know, in a hectare you would have 100,000 times bigger. And I wanted to make it happen, so I sprayed with 2 liters. I didn't incorporate, and I water as much as possible to see whether the herbicide would have bleached the lupin, and the lupin bleached, and the Overwatch symptoms are there. I'm still working on this. I definitely had identical, or what I believe are kind of identical symptoms to the field, and yeah, this is something that everybody could do, because with this app, the Canopio, you can measure the green. And yeah, I'm still trying to quantify how much green there is. This is plants 3 weeks after. It seems that the lupin is coming back. Yeah, I'm just working on it. I don't like to work on it because I prefer to focus on herbicide resistance, but I just wanted to learn as much as possible because I felt a sense of duty that, you know, I'm paid by you guys, basically. You know, you pay the levy, and GRDC decides that it's good to invest in Ubeki, and then Ubeki hires me. So eventually I get a salary. And so I feel that, you know, and I've seen how good Overwatch is, and I believe that if I can provide some data that will allow this herbicide to be used in the future, is a good thing. And yeah, Clethodim on its own, we mentioned already of the 34%, and this is the other mix that I've used that applies to loopings only. When it is mixed with Factor, resistance basically decreases by 3-4 times at least, and it goes down to 8% only. Okay, so radish, I will follow same sort of pattern, starting with the knockdown. Yeah, we didn't find any glyphosate resistance in radish. The pattern that I used was 2 applications of Crucial. Of course, with only one, you tend to suppress radish more than killing it. And I've seen that 2 applications 10 days apart, trying to replicate what you would do in Roundup Ready canola, resulted in full control of radish. And also last year I didn't find any glyphosate resistant radish, so glyphosate keeps working. And then yeah, this new chemical, Group G, Terrad'or, I use it at its lower rate. I've introduced a sample that in 2020 had a couple of plants surviving, and so at 20 grams it was good to confirm that there seemed to be some very, very low level of resistance. I mean, I sprayed it again with 20 grams and those plants were killed. Last year I tested Voraxor, not on in a pre-em sort of use pattern, but post-emergent at 100 mils, and everything was killed. So, and I this year, together with Terrad'or, I also tested for Reflex, and yeah, the overall result that I consistently confirm is yeah, basically no resistance to Group Gs, at least the ones that I tested. Reflex I tested at a very high rate. On a subsamples I tested also a liter, but still it's very powerful. And again, the loading of active ingredient that you put, it's an order of magnitude. It's like 200 grams or 250 versus you know, the Voraxor or the Terrad'or is only 20 grams or 37 grams, 100 mils. So, so no resistance there. And again, I try to replicate the use pattern of, I don't even know how to start it, but maybe of those, this is a video from Richard Quinlan Plant Farm, and this was Reflex used as in a sort of knockdown situation together with glyphosate. And this video Richard took a few days only after Reflex application, and you can see how effective it was on very large radish. So, and again, today Roger has mentioned the comparison between Calisto and Voraxor, and so we do test, but there's always more that we can do using Reflex not as a pre-em but as a post-em, or vice versa, using Voraxor testing as a pre-em and post-em. So yeah, it works really well, no resistance there, and it's very active on big radish. Video keeps on going forever. So with last year and this year I tested for Calisto standalone at 200 mils. I tried to have a quite sandy potting mix. This was a sample that I identified last year that I used as a reference, and yeah, that sample had about 30% survival. And overall, I repeated this with potting mix and with sandy soil, about, you know, in the range of 10% or a bit more of the samples had a level of resistance to Calisto, even though it has been introduced only in 2020. And it's a standalone pre-emergent. So this was the figure. The interesting thing, we have spoken about Overwatch. Overwatch has also activity on the radish. The label claim is only for 60 or 70% of control, more like suppression. But when it was used with together with Calisto, the same sample that you saw before was fully killed. So we went from 30% survival to 0. And again, this is an area where needs probably a bit of fine tune because again, the Syngenta strategy is to use it with boxer dog, of course, and yeah, sure enough, Prosulfocarb does offer some suppression of radish, but also Sakura does offer some suppression of radish, and also Overwatch. And so, yeah, it's probably good to know which strategy is better, so you have full knowledge of the technology you are using, not just to manage resistance, also, of course, to control weeds. And yeah, the same photo. Originally I didn't test for Overwatch standalone, and then when I put this on Twitter, the agronomist told me, well, but have you tested actually Overwatch on its own? And so I went back and I did, and then this is the final snapshot. The Overwatch didn't allow many ryegrass plants to come through, and I saw that it performed similarly to what a Group F would do. Sometimes populations that were triflufenican or picolinafan resistant were also I could see greater level of survival to Overwatch. But yeah, the good story was this one, that it helped to manage HPPD resistance or group H.
Sorry, Roberto, that Calisto resistance here, was that a field evolved resistance or was that a glasshouse field?
Yeah, it's a population. Last year I identified 2 populations that were resistant to velocity, and
from the field.
Yeah, from the field. And one of them, that particular one, it's a farm where basically there are no weeds, lots of harvest with seed control, and that radish was just collected on a patch that was missed. Because again, this is what I've learned in the workshops. It's difficult to demonstrate, but yeah, Garen was the facilitator in one of the other workshops we took around the state, and he submitted. He collected samples for 2 farms. One was a very not very proactive farmer, and there was lots of ryegrass in the farm, and one was a very proactive farmer. Garen spent 2 hours to take a sample out. Which farm had more resistance? The second one. So the guy that does everything right had a lot of resistant Trifluralin and some of Overwatch, while the other guy, maybe it was just random chance of picking patches that were actually missed. But still, it tells a lot that sometimes because of the system is so much optimized with harvest with seed control, the numbers of weeds are low, and sometimes you may think that you don't realize that there is a problem in the background, and it's good to know maybe if you think that's useful and helping the business. Yeah, Nick mentioned before, this is one of his samples, and this Atrazine was only found in 4 samples. One was my resistant control, and Nick was one of those, but that one was susceptible to Logran. So again, when you have a comprehensive list of stuff that is tested, you're definitely gonna find something that works, whether it is a mix or a standalone, and then you find your way of trying to make that an economical choice that doesn't kill the crop. And so all of those guys were then sprayed with Diron. I went very high because Atrazine I sprayed twice, 1.1 kilos, and I read the Adama label. The first number was 1.1 Diron. I know that is a high dose, but actually I didn't know. I got told in a subsequent workshop. So I sprayed 1.1 kilos. They were developed plants. It took forever for them to die, but all of them died. So again, same type of, now they are actually split in 2. It will be group 5 and 6. Before they were all under the group C.
So Diron and Atrazine split?
Yeah.
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 It's going to get confusing.
It is. The only one that knows everything is you, Becky. But we are still learning, and the curve is extremely flat, but we think it's steep. So yeah, not much surprise here. Similar figures for Logran and Intervix. Generally, the imidazolinone works a bit better than the sulfonylurea. It provides a bit a greater kill, but again, I only test small samples for radish. I put in pots 20 seeds, And so sometimes I had 15 seedlings coming through, and that's why probably, you know, the discrepancy between, you know, one out of 15 already, we are almost into the category of developing resistance. But again, overall, 70% of the samples had some ALS resistance. And again, 30% is still up for grab, and Nick was one of them. Same identical story for 2,4-D and MCPA, up in the 50% or greater of the samples submitted had some resistance, and it is the similar story for diflufenica. I have also tested picolinaphen, but the results aren't great, so I only presented Brodal. And yeah, this was early on. You could already see which samples had plants recovering and growing through. This one is fully bleached. There wouldn't be no new growth, while, you know, the one that are resistant produce another leaf, and away you go. So 50% is a very high number, but when you mix it with bromoxynil in the form of Jag or Quadrant, which has also the picolinaphen and the MCPA, basically the resistance goes from 50 to 5. And so that tells me that the group C, bromoxynil and atrazine, but bromoxynil especially, is amazing. You know, is one of the best herbicides we have. Of course, good herbicides are banned in Europe, so bromoxynil is not available to be used in Europe, while here luckily we have, because it keeps alive not only the F but also the H. And yeah, almost identical sort of pattern between the Quadrant and the Jag. And so last year I identified this velocity resistant radish, and I did subsequent work to understand whether, you know, the resistance would have extended to the frequency, to the Calisto. And so yeah, this was the test. If you do test with me, you get back a table. You don't get back a report. We are trying to make everything efficient, so it's difficult for me to write every single report. I assume that you can read and numbers and understand that 60% survival to Logran is bad, and so sometimes I highlight in red or yellow or green. So, but this population, what I found quite consistently is that radish struggles to keep F and H together. So most of the time, if you are HPPD, group H resistance, you are not F. And only one population from this year had a level of resistance to group H, and it was resistant to group F. I don't know whether that is reflecting, you know, a lot of looping grown versus a lot of wheat, or whether it is actually very difficult for radish to hold those together. But on that
in our lupin growing area, we've used a lot of group F and a lot of group H. So it's sort of not like they're mutually exclusive. So it sort of suggests to me that the plant struggles.
I've seen this as a, you know, something that I don't have yet published the paper. I will, with the stats, get a better insight, but I see it quite consistently. So that tells me that if a company comes up with an F and H together, it's a good strategy. But yeah, definitely, you know, the bleachers have same symptoms, but they are different. I believe that HPPD get detoxified in a way and DFF in another way. And so I saw that quite consistently, to the point that this year I want to make crosses from that particular population and see whether they are, you know, segregating or whatever they are. But on that particular population, I then tested the cross resistance, and yes, it was resistant to Calisto. It was resistant to velocity. When I spray to 4 leaves, you know, big plants, you know, the efficacy on this are 4 reps of the same. This is my susceptible. It wasn't killed. I went to velocity plus MCPA, same story. I went to a liter. This was a study I did last year to confirm it, same thing. I used frequency plus bromoxynil, 900 mils of bromoxynil, same story. Frequency and MCPA for leaves even worse, because you can see the biomass there, and clear symptoms of MCPA efficacy there. What I found, and was really interesting, and then I followed this thing up, and I'm still studying to confirm again with the sample from this year, is that when I use frequency as an off-label, it was actually more effective on this same population, was more effective than when it was used with MCPA. Of course, this is cherry picking, but it seemed that MCPA is actually antagonizing the efficacy of the topramezone. But all still to be confirmed. The interesting thing is that the same population, but now sprayed small, was almost fully controlled with 500 of velocity. And when I went to 670, it was fully controlled, 100%, and a liter also. So I think that many times when you see something that doesn't work in the field, it's probably because you sprayed big radish or a different size radish. And when that was tested in the lab, it is tested at this size and resistance isn't found. Maybe tested in March with, plants still quite soft in a glass house, high light intensity, and the plant completely killed. And the glass house is tricky. This is my result from this year. I found 2 populations last year. I thought I would have found a lot this year. This is what I found. I sprayed all of these 96 samples, and this is my resistant control. 1 plant survived, 670, and this saved my reputation because at least I could confirm. And this was mid-March, so the temperatures start to drop, so the velocity is very effective, but the light intensity is high. And I'm not presenting. I redid the same thing now, sprayed on 24th of June. We were in a lockdown, or maybe 29th of June. Same rate, full of radish, same size in June. So I'm now tidying it up. I went back with a liter, etc., but I don't have time to cover. What I found quite interesting, because I had this feedback that, in my opinion, the MCPA was antagonizing, this year one of the testing options was velocity 670 plus MCPA. I sprayed the same day. Same dose, 670, but then I came back with alpha liter, 500 mils of MCPA, and I found this. So I was told Bayer, they were very interested. I went to do all the workshop. I said, this is a true story. This is the big deal, big discovery of 2021. I repeated and I couldn't do it. So, so in winter, actually in winter, MCPA seems to be adding, because again, I receive lots of feedback from companies, not just Bayer, that MCPA in the field seems to add something rather than detract something. So yeah, you either do the experiment only once, or then you have to do it 3 times. You never repeat the experiment twice, because you have one black and white, and then you need to. So I'll repeat it again, and I will decide which. But this was very real because well, I sprayed on the same day, and the plants got the same solution from the same bottle. I just added the MCPA, so the interpretation is quite easy to be made. And again, the important message that I wanted to convey is that sometimes when you see that the velocity or HPPD has not performed the way you would have expected, maybe think about whether you have added the MCPA. But at the moment, I couldn't confirm this, and I'm not hiding, the results. I'm telling you exactly my successes and all the failures. So yeah, 10% of the samples had some level of survival when I had the MCPA. These are the figures. When frequency is used with MCPA, 20% of the samples are resistant. Calisto, 5% within the GRDC sort of samples and the velocity 41.0. Here's the brome that Nick submitted for testing. So identical response. It was the one that was most resistant to Clethodim, and applied 250 mils, and Fusilade 250 mils, same sort of response. It is pretty much all you have to see as far as group A resistance is concerned. I couldn't kill this brome with a mix of butroxidine and Clethodim. Then I learned that maybe the factor is not as good on brome, so I tested Clethodim plus Quizalofop, QPE, and I still can't kill it. So sometimes the mix works with ryegrass, but the same thing with another species doesn't work. So it would be interesting probably to dig into the DNA and see what type of genetics this thing is, and maybe do a dose response to clarify the issue.
So do we get one brome resistant population and 49 susceptible ones, sort of thing? Is that what I'm reading?
Yeah. There was only this one that was clearly
Yeah.
resistant. Maybe another one. This one was also resistant to Group B. Overall, pretty much 10%. I've used the sulfurmeturon, which is not used, is for industry. I've used to, and it's quite harsh. On the brome, I use Monza, but Monza is very difficult to use in post-emergent. It's very much size dependent. Overall, it's about 10% of the samples had some level of resistance, and it's pretty much it. I tested Intervix, and one of the population that Nick submitted had some plants that were clear. Actually, those are the plants. You know, there was a clear difference. These have received 750 mils of Intervix. I contacted BSF in Germany. We were ready to go for a project, and then I transplanted those guys to get the seed, and they died. So, yeah, even when you see a clear phenotype, I mean, the IMI sometimes don't kill grasses that well. I mean, definitely it was effective in the pot next door, but yeah, again, it's the beauty of biology. I thought I had a clear phenotype, but at the moment I can't confirm these results. So yeah, no IMI resistance has never been reported. It would have been a first. It has been reported in barley grass, but not in brome grass. And yeah, I tried a few pre-em herbicides because I wanted to learn how they would work. We talk about Ultro that can be used in lupins, and it worked really well. 1.1 kilos, I tried propizamide, works 100%, and also 3 liters of Avadex worked 100% on brome. So that was probably good to share. I tested Luximax, and for a while it worked well, but for no reason some of the samples were not fully controlled. I wouldn't call for resistance, given that is only suppression claim on the label, if I'm not wrong. Definitely Overwatch is not for brome. In fact, those plants are all red because at that time, 3 months ago, were sprayed then with Intervix. And the population I showed you before is coming from all of this bunch. So yeah, this is it. I think yeah, the key, we didn't go through the key messages before, but it's just basically a summary that basically the F and H are, in my opinion, under pressure. The group F much greater pressure because there is more resistance, and they are all kept alive because of the bromoxynil. Atrazine works really well. Only 2% of the samples have resistance, and there is no resistance to G and M. And yeah, basically mix, because by mixing you can get effective control of radish. Ongoing research, I mentioned that well, we moved to the padding to get into the real world and really understand a bit better the compromise of growing a crop and killing weeds We are doing a field experiment in collaboration with University of Adelaide, focusing on pre-emergent herbicides, and the idea is just to safeguard group K, because with Sakura going off patent, I think that Sakura resistance will potentially increase significantly. And we study the physiology of resistance very much. I collaborate a lot with Dr. Danica Gogin. She basically tries to understand at the fundamental level what I see in pots.